Okay, so here we're showing you the contents of one of those foils when we've opened it. We've, in this one, got two little sachets of desiccant, which are just pouches to absorb any moisture that could have entered into the foil. Those ensure that the test membranes are kept dry at all times prior to you doing the test. And here we have the actual test cup. We've got on here an area for putting the ID of the test, the date, and the results recorded by on there. You'll see, looking in from the back, you can see the test membranes embedded into the side of the cup. And you've also got a temperature test strip on the side here, which is a little bit of chain of custody. You should be getting a urine sample put into here which shows between 36 and 37 degrees at normal healthy body temperature. If it's well below that or not showing anything on the scale, it would tend to indicate that it could have been adulterated or diluted or substituted with a sample that's not come from a body. Okay, and to perform the test, you basically unscrew the lid and your sample goes into there. So this can be given directly to the person that you're wishing to test. If you can do a semi-observed sample collection, all the better, but obviously if urine testing, that can be problematic. And when the sample is applied into the container, we're just going to add some liquid off camera to this. The lid can then be reapplied. And at that stage, it's completely waterproof. You're not going to get any dripping or any problems with contamination. Now obviously, depth of the sample you're looking to gain is at least over this temperature strip. If it's below that or insufficient, there's a minimum line on here. Anything below that and you're not going to be able to run the temperature strip. The fluid that we've put into this for demonstration purposes is not at body temperature, so I'm not expecting the temperature strip to come up and show on this one. That's perfectly normal. But you would expect to see green discoloration on this liquid crystal display at around 36 to 37 degrees. Now, the read time for these tests is in the data sheet, and it's very important that once the sample has been applied into there, you start a timer and note the duration until you read the test results showing. There is a recommended read time of between 5 and 10 minutes on these tests, and to read your results, what you do is just peel this test membrane away here. The label comes off, and what you'll see here is your 1, 2, 3, 4, 5, 6, 7 test membranes with the coding on there, so you can interpret which results you're looking at. And then you're looking for the presence and absence of control lines on each of these membranes. The control lines form at the top of the test membrane, and the test lines are those showing about 5 to 8 millimeters below those. Now you'll see here we've got an all negative test result on this sample, showing test lines and control lines at the designated read time. Now these will continue to develop until around 10 minutes. If there is dye still showing, pinkish dye showing on the white test area, you should wait for the read time, and the membrane will normally clear completely to give you a white background. The test lines will vary in colour density, thickness and saturation. If we just bring that close to camera, you'll see there's quite a variation between the OPI membrane test thickness and colour density and the THC test line. This is quite normal. Any test line showing in the presence of a control line at the specified read time is a clear negative result. You must not try and interpret anything by the thinness or paleness of the test line. Particularly the very sensitive membranes such as the THC, they tend to be thinner and fainter than the test lines that are working at higher levels of detection. But this is an all negative test, and actually as soon as you've got an all negative test and you've got two lines showing clearly on each test membrane, you can interpret those as clear negative results across the 7 that have been tested and move on to the next test if you wish to do so. So that's the UK Drug Testing 7-in-1 cup collection system. It's a very good value product. The accuracy of these membranes is all between 98 and 99% on these 7, which makes it a very accurate test compared to a gas chromatography mass spectrometry laboratory test. They're available from the UK Drug Testing website, can be purchased individually or in multiples of packs of 10. We do recommend you buy 10 as a minimum starter, ideally, because then you get the full data sheet for your reference. It gives you a lot of information in there in terms of cross-reactivity information, You get full technical support, obviously, in the UK during office hours on these if you run into any problems with them or difficulties with the interpretation. But there's really very little that can go wrong with the procedure in terms of the collection and interpretation of your results with this test. It works very well, it's accurate, very selective and very sensitive.